We have to start this session, and we have the pleasure to welcome Professor Abdul Barakat for a plenary session. So this talk will be devoted to microstructured surfaces for controlling cellular behavior. I would like to introduce just by a few words Professor Barakat. You obtained your PhD, I believe, in 1996.
1994?
1994. So
I'm even older than you think.
Everything is increasing.
Yeah.
1994 in the Massachusetts Institute of Technology.
Right.
True?
Right.
Okay. After that, you become postdoc at the National Institute for Health in Chicago,
Yep.
I believe.
Yep.
Then assistant professor at the University of California. And in 1996, is that what? That's okay.
95, I started at the University of California.
Okay.
Yeah.
Then assistant professor in the same university.
Yeah.
And then full professor
Yeah.
in the same university too in 2006. And after 4 years, in 2010, you came in France for becoming director of research at the CNRS, National Center for Scientific Research, and both full professor at the École Polytechnique in France. And in the same time, you are professor too at the University of Sydney, adjunct professor at the University of Sydney, and also principal investigator in the Center of Bioengineering in China, in Beijing. And then we are very pleased to welcome you and to hear your talk. And after your talk, I will have the pleasure to give you the prize Eugenio Beltrami, of course, with the committee and on behalf of the Mathematics and Mechanics for Complex System Research Center. So please, Professor, it's your time.
Thank you very much. Thank you very much for these kind words, Yves, and thank you all for the invitation. I'm delighted to be here. So I'm going to tell you a little bit about microstructured surfaces. And most of what I'm going to tell you today is going to be experimental. But I hope you'll see in the experiments that I'm going to show you opportunities for doing mathematical modeling and computational modeling. We're interested in these microstructured surfaces for a number of reasons, as you'll see, but these reasons really range from understanding basic cellular processes and then going potentially all the way to diagnosing certain diseases. So that's kind of the realm we're talking about. So as Yves mentioned, I'm at École Polytechnique in France, and I'm in a laboratory called the Laboratoire d'Hydrodynamique, so the hydrodynamics laboratory. It's a bunch of fluid dynamicists, basically. This is my group today, and Manuel actually does have a body, even though it doesn't show. It's not just a head and a neck, but he just wasn't for the photo that day, so we've added him on. But most of the work that I'm going to present to you is the work of Claire Leclèche, who's a postdoc in the lab. I'll also allude to some earlier work from a former student called Josh Morgan. And these are the funding agencies that generously fund our work. So let me just motivate what we're interested in. And really, most of what I'm going to tell you about is motivated by our desire to understand atherosclerosis. So atherosclerosis is the disease that leads to heart attacks and strokes, which are the leading cause of mortality in the world, right? And this is what atherosclerosis looks like in its early stages. So this is a picture taken by fiber optic lighting of the inside of an aorta. The aorta is the largest artery in our body. The aorta of a rabbit. And rabbits typically don't get atherosclerosis spontaneously, but if you feed them what we eat, then it's very effective at basically getting the disease. And so what you're seeing here, this is the arterial wall, and you see these bumps here. These bumps are the initial deposits of fat, of lipoproteins, of blood cells in the arterial wall. And then in the advanced stages of the disease, these deposits grow until they form plaques or lesions that can protrude into the vessel lumen and potentially block a blood vessel, right? So if this were to happen, for example, in the coronary artery, it can cause a heart attack. Alternatively, this mass may get detached under the influence of blood flow, travel to the brain, and cause a stroke. Right? So that's the disease we're interested in. Let me just say a few words about how the disease develops, and at the same time kind of talk about the system that we're studying. So this is what a normal artery might look like. So up here is the lumen where blood flows, and then this is the arterial wall. So this monolayer, single layer of cells that lines the inner surface of all of our blood vessels is called the endothelium, right? So it's endothelial cells. I'm going to talk a lot about endothelial cells today. And so that's the green structure here. And then further down within the arterial wall is another cell type called smooth muscle cells. And these are the cells that allow a blood vessel to contract and dilate. So these are the muscle cells in the arterial wall. And then further out, there are other cell types like fibroblasts and mast cells and immune cells that we're not going to be concerned with today. Now, this endothelium that I talked about, this green structure here, is very, very important to our health. So it serves a number of functions, and one of its major functions is to regulate transport, to regulate permeability. So typically anything smaller than 4 nanometers in hydrodynamic diameter doesn't get through the endothelium, right? At least not in between cells. It has to have specific receptors on cell surfaces that would internalize that molecule. So it's kind of a gatekeeper, a very effective gatekeeper at keeping away things like cholesterol. Cholesterol is carried by a lipoprotein called low-density lipoprotein, which has a size of 22 nanometers. So it doesn't get through very easily, right? So permeability regulation is one of the major functions of the endothelium. The endothelium also regulates smooth muscle cell proliferation. These other cells that I was talking about here, if you have a dysfunctional or a damaged endothelium, these cells start proliferating and migrating in an uncontrolled manner, and they can actually invade the lumen and block it, right? And so the endothelium, when it's functioning properly, releases some molecules, some factors that act on smooth muscle cells and keep them in a quiescent state. And then another very important function to the endothelium is what we call anti-thrombogenicity. What that basically means is it doesn't allow clotting, right? It's a natural anticoagulant. The reason blood doesn't coagulate spontaneously in your blood vessels, and it does when you put a drop of blood on the surface, is because the endothelium actually expresses anticoagulant properties. Okay, so that's the endothelium, and keep that in mind because we're going to be talking about endothelial cells pretty much throughout the talk. So what happens in atherosclerosis? Well, the atherosclerosis, the disease, is triggered by chronic inflammation of the endothelium. That's what happens, in fact. That's the trigger event. That chronic inflammation can occur in response to various things, what we call oxidized molecule. So certain molecules in our bloodstream that can get oxidized can actually cause inflammation in the endothelium. You can cause endothelial inflammation by viral infection. Certain radiation can cause it. Certain bacterial infection can cause it. It turns out that certain mechanical factors also can cause it. Certain types of blood flow, because you can have of blood flow inside your arteries. Certain patterns of blood flow can actually induce endothelial inflammation. So when the endothelium is chronically inflamed, then it can't do this stuff very well, right? So for example, its ability to regulate permeability is compromised, and you start getting massive influx of things like cholesterol into the arterial wall and where it starts accumulating. You also get an inflammatory response, which means blood cells, white blood cells, in this case monocytes, can adhere to the inflamed endothelium, so it becomes sticky, and then they go into the arterial wall, and so over time you start getting accumulation of cholesterol, accumulation of these transformed white blood cells, and this is what we call the atherosclerotic plaque or lesion that is developing over time. And I mean, atherosclerosis is not something that happens over months, right? Usually you don't see too many 15-year-olds who die of heart attacks, right? So it's typically a disease that happens over decades. So the real risk in atherosclerosis is that this plaque ruptures. Okay, so there's a whole field of study about what we call the vulnerable plaque. People are interested in trying to understand when a plaque becomes vulnerable to rupture, right? So when the plaque ruptures, then the continuity of the endothelium is disrupted. And as a result of that disruption, if you think of the fact that it's an antithrombogenic surface, if you don't have the endothelium, all of a sudden you get a very rapid development of a clot, and that can be a heart attack in a coronary artery, for example, right? In fact, if you think about it, atherosclerosis is probably the only disease. Oftentimes, the first symptom is sudden death, right? Okay, now it turns out that atherosclerosis doesn't occur randomly in our arterial system. It actually is a focal disease. And what I mean by that is the lesions don't develop everywhere. They develop specifically near branches and bifurcations. Number one, it's an arterial disease. It doesn't happen in veins. It only happens in arteries. And in arteries, it doesn't happen everywhere, right? If you think about it, when you go, let's say, to a doctor to get evaluated for your risk of cardiovascular disease, what would he or she look at? They'll do a cholesterol test, right? They'll check your blood pressure. They'll check if you're aging, because these are all risk factors, right? High cholesterol, hypertension. If you're a man, you're more likely to get the disease. Sedentary lifestyle is a risk factor for atherosclerosis, right? There are all these risks: smoking, diabetes, all these are risk factors. But there are some people who have all of these risk factors and never a problem. And there are other people who have none of these risk factors and all of a sudden keel over and die of a heart attack, right? So we think our understanding of risk factors for atherosclerosis is incomplete. And part of what we think is missing is mechanics, it turns out. So, and that link to mechanics is really provided by this observation that if you have high cholesterol, you have high cholesterol everywhere. If you're a man everywhere, presumably. If you're getting older, you're getting older everywhere, again, presumably. But it's only at these specific sites of branches and bifurcations that you're getting the disease, right? So over the past 40 years or so, people have, so this observation of this focal nature has been known. It was actually first made by a Russian scientist, Anechkov, back in 1910. But then since the 1980s, people have tried to link it to flow, to fluid mechanics. The idea is if you look at using various modalities, you can kind of visualize what the flow field might look like around arterial branches and bifurcations, and you would see things like the development of flow separation, boundary layer separation, and flow recirculation. And these regions of what we call disturbed flow, even though the exact definition of flow disturbance isn't that well documented, but these regions of disturbed flow are typically the regions that are most prone to atherosclerosis, right? So there's a whole interest in trying to understand in detail arterial, specifically the wall shear stress, right? The frictional arterial wall as a result of blood flow, how that shear stress changes in time and in space. So all these issues are important. Now, another really interesting thing about atherosclerosis is shown here. So if you actually, this is a section of an aorta, again, section larger than our body, that's been cut open and pinned flat down on the endothelial surface, right? So the endothelium is this monolayer of cells that lines the blood vessels. And this area, so each one of these contours here is a single endothelial cell, right? And this happens to be just around a branching area. So this is just upstream of the branch, and this is within the branching zone. And what you can clearly see is a transition in cell shape from this zone left of this yellow contour to the right of it, right? So in here, the cells are elongated, all nicely aligned. And then within just a few cell lengths, you get the situation where the cells are round and randomly oriented. What's really interesting is that these cells here, or this arterial zone here, tends to be protected from atherosclerosis, whereas this zone here where the cells are round tends to be prone to atherosclerosis. So what we and a number of other groups around the world are interested in is trying to understand this link between cell shape and function, the cell shape and predisposition for the disease, right? Does shape in itself cause predisposition to the disease? And what factors regulate endothelial cell shape?
So the shape of what?
Of the endothelial cells, of these individual endothelial cells, yes. So you see how these cells are elongated and aligned, and these cells are much more round. And the fact that these cells are prone to the disease, whereas these cells are protected from the disease, is what we're interested in. Why are elongated and aligned cells more protected and round cells are more prone to disease? And then what, even upstream of that, what regulates cell shape in our bodies? Okay. So yeah, the idea is this zone here corresponds to this region of flow disturbance. Now, if you look in the endothelial cell literature, you would find quite a few papers that talk about how you can regulate endothelial cell shape by flow. So there have been many studies, typically in vitro, so on cultured cells, where you take endothelial cells. This is work we did back in 2000, you see. So that you take endothelial cells and you culture them in the laboratory, and you form monolayers, and then you put them on here. So we're exposing them to flow for 24 hours. So this is 0, 2, 6, 12, and 24 hours. And first we put them under an undisturbed flow. So a flow would be where you would get a protected zone in our arteries. And you see you can go from round cells to highly elongated and aligned cells. The flow is in the direction of the arrow here. But if you take the same cells and put them under an oscillatory flow, so a flow that just oscillates back and forth for a period of 24 hours, then the cells stay round, right? So you can certainly control endothelial cell shape by applying different types of flow, okay? But this is not what I'm interested in, what I'm going to tell you about today, because there's another story to this that I think is even more interesting. So a few years ago, one of my former PhD students took some of these endothelial cells in an artery, and he removed the endothelial cells, and he imaged the basement, what we call the basement membrane. That's the surface on which the endothelial cells sit in vivo. Okay, so. And this is what it looks like. This is an electron microscopy image of what it looks like. So you can see it's not at all a smooth surface, right? It's a rough surface. And this roughness, depending on at what scale you look at, can be either at the nanoscale or at the microscale. Okay? So we started getting interested in this roughness of the basement membrane, or on the substrate that the cells live on in vivo, and wanted to understand if that had any effect on cell shape. So just to show you, it turns out that all of our cells, adherent cells in our bodies, sit on rough surfaces. There's no such thing as a smooth surface that cells sit on in our bodies, right? So these are just various examples from veins, and this another vein. This is a carotid artery. This is a descending aorta. But it turns out it's also true for a lot of other tissues. The basement membrane on which cells sit are typically topographic surfaces. They have some roughness associated, and they're microstructured and nanostructured surfaces, right? So this is what the topography looks like. This roughness of the surfaces might look like in various tissues. And to study these things in vitro, so now in the laboratory, we use different systems. We can use different systems. Some people work on rough surfaces. Some people put fibers together on which they culture cells. Some people make pillars, micropillars or nanopillars on which they culture cells. And some people use these arrays of grooves, microgrooves. And this is, in fact, the system that we've opted for. And that's what I'm going to be telling you about most of the day today. Okay? So we take these surfaces, we fabricate these surfaces at either the nanoscale or the microscale, and we put cells on them, and we try to understand what the cells are doing, endothelial cells. This is just some example about what cells do. This is from different studies. What cells do when put on these grooves, these microgrooves, right? And what we've learned over the past 10, 15 years, is that there's a process called contact guidance when cells are cultured on microgrooves, where basically the cells in their orientation and in their organization are guided by these grooves, right? So you often see the cells are highly elongated and aligned in the direction of the grooves, right? So that happens in many, many cell types. The thing is, there are several gaps in our understanding. One gap is that we don't know, there's very little work on if you have both the flow on their apical surface, on the top surface. So what happens if you have these combined signals, the signal of the grooves plus the signal from the flow? Most of the contact guidance studies in the literature focus on single cells, but endothelial cells live in monolayers, behave collectively. So it's important to collective of endothelial cells might be to the grooves. And then we want to try and understand how this contact guidance happens, right? What is actually happening to the cell in the direction of the... So these are three things that we've been thinking about and been trying to address. Let's start with the effect of flow. So we started with, in this case, nano grooves. So we made grooves that are that have a pitch, so spacing between ridges. So the grooves, basically you have a groove here and you've got a ridge, right? A groove, ridge, groove, ridge. So if the spacing between ridges, between the half point of the two ridges, is what we're calling the pitch. And we are going to vary it from anywhere from 400 nanometers to 4 microns. And then in this case, the ridge height is 300 nanometers. So that's the structure we're looking at. These are the 6 surfaces we're making at 6 different pitch values. And to make sure it's clear, a single endothelial cell is something microns in diameter. So we're basically, one of these endothelial cells is going to be sitting on multiple ridges and grooves, right? Okay, and what we're going to do is we're going to culture endothelial cells on these grooves, and we're going to put flow on top, right? And so first, we're going to have the ridges and the grooves in the same direction as the flow. And this is just for 2 different pitch values, 800 nanometers and 2 microns. And what you can see is the cells that were initially round if you put them on a flat surface. On these combined flow and microgroove surfaces, the cells become elongated and aligned in the direction of the flow and the microgroove. So that's contact guidance because of the microgroove, but it's also elongation of the cells because of the flow, right? But really the more environment happens or in orthogonal directions. That was what we were interested in. Like how you have now the grooves are basically coaxing the cells to elongate in this direction, and the flow would be pushing them to elongate in. And so the question, what happens, right? And what we found is at a pitch of 800 nanometers, the cells stay more or less round, and then if you put them under 2 micron pitch, then it's actually the grooves that win and not. So this to us was a very interesting observation because it basically showed that from the bottom, from top, the flow information, and it's that integration between the 2 types of signals that ultimately determines what cell shape and cell orientation looks like, right? And it turns out that under certain conditions, it's the grooves that win, and under other conditions, it's the flow that wins. And it really depends on the architecture of the grooves. It depends on the spacing between the grooves, depends on the height of the grooves, and so on. So it turns out this is not true only for alignment and elongation of the cells. Turns starts integrating the information, and they migrate both in the direction of the flow, but also in the direction of the grooves. And that integration, how that happens, is not understood, and something that we're interested in. Okay, now I want to get to some recent work where we saw that these grooves were interesting, so we actually backed up and wanted to just understand what the cells, our cells, do on the grooves without even the flow, right? So we make, in this case, micro grooves. So these are these ridges and grooves that I was talking about. In this case, the ridge height is anywhere from 1 to 6 microns, okay? And we culture cells on these surfaces. Here, we wanted to start to understand the response of monolayers of cells. The question we wanted to ask was, okay, we know if you put a single endothelial cells on these grooves, you can elongate the cells and align it. The question is, can you also do the same thing to a monolayer? Because it's not an obvious answer. I mean, because the cells interact through junctions between cell-cell junctions, and you can imagine that those junctions, the forces between cells through junctions might outweigh the effect of the grooves, and the cells might remain round, for example. But what we found is if you put cells on flat surfaces, so no grooves, and you wait, say, 48 hours, the cells, so here you're looking at each individual cell, so this we do some segmentation to get individual cells, cell contours, and then in color is the orientation of the cell, right? So you could see the cells on flat surfaces remain randomly oriented, whereas if you put them on the microgrooves and the groove direction is horizontal, you see how they become all aligned and elongated, right? So we now know that with a monolayer, just like with single cells, we can get elongation and alignment in the direction of the grooves. And this is interesting again, because that means if I culture cells on flat surfaces, I can get randomly elongated cells, that kind of cells that I showed you earlier that are prone to atherosclerosis, whereas I can now non-invasively control endothelial cell shape and orientation and by the grooves and make them look like the protected cells up here, right? So the next step, of course, is to take these two cell configurations and probe their function, right? And see if this is indeed more prone to inflammation, which leads to atherosclerosis, and this would be more protected from inflammation. Okay. Then we started looking a little bit more at how changes in the architecture of the grooves might play a role in determining what's happening to the cells. The groove depth is the primary factor, the primary parameter that affects the extent of alignment. For example, this micron deep groove, you get even more elongation and alignment, right? It turns out if groove width and groove spacing doesn't have much of an effect. It's really the depth of the grooves that's most interesting. Then we started looking at things like the mechanisms, the biological mechanisms that actually are driving this elongation and alignment. So there's a theory out there for contact guidance that's called the focal adhesion-based theory. The idea is cells, when they actually sit on a surface, they don't sit flat on the surface. They actually have kind of like feet, right? What we call focal adhesions. They make adhesion with surfaces at specific discrete points. And so each one of these dots here is one of these focal adhesions that the cell uses to make attachment to the surface, right? And the focal adhesion-based theory says the way cells elongate and align on grooves is by elongating focal adhesions, which then organize the cytoskeleton, primarily the actin cytoskeleton in cells, and that's what actually causes cellular elongation and alignment. Well, we found that that's not what we're seeing. It's with polylysine, which doesn't allow the formation of focal adhesions, and the cells are just equally elongated and aligned. And we also can disrupt the actin cytoskeleton, and they still remain aligned and elongated. So we think, rather than this focal adhesion-based theory, what we're proposing is that cells actually align and elongate on microgrooves through protrusions. So what we've done here is we've looked at how the cells what kind of elongation, local elongation in the cell happens as the cell sits on the microgrooves. And we found that the cells makes these protrusions right on the ridges of the grooves, right? And some of these protrusions are also in the direction of the grooves, and some are actually go into the ridges and into the grooves themselves, right? So that you start on the ridge on the top surface, and you either elongate in the direction of the groove, or you could go inside the grooves, right? And so in blue here are the protrusions that go axial, and in yellow are the protrusions that go into the grooves. And so we can visualize these protrusions in live cells. And what we found is the axial protrusions have a longer lifetime than the lateral protrusions, and they have a larger total displacement than the lateral protrusions. So what we think is happening is that the physical constraints by the grooves on cell protrusions are the main drivers of contact guidance response, as opposed to the focal adhesions and the organization of the actin cytoskeleton. Okay, the other thing we found with these groove systems is if you let the cells proliferate sufficiently long on these groove surfaces so that the cell density becomes sufficiently high, then you lose the effect of contact guidance. You lose the effect of elongation and alignment. This is seen here. Are elongated, and by the you've basically lost the elongation and alignment. And we wanted to understand why that might be happening. So this is just to quantify how you lose both the alignment and the elongation with cell density. And what it turned, just to kind of summarize the story here, what you find is that these cells release and secrete their own basement membrane. They secrete their own proteins that actually end up masking the grooves. So this is the top, looking from top, and this is looking from the side. So you see these rigid grooves, and you see all these proteins. These are fibronectin, collagen 4, laminin. These are all types of proteins that the cells secrete that end up covering the groove surface and end up basically masking the grooves, so the cells no longer see the topography. All they see is this layer of secreted proteins, and that's what they end up responding to. So we show here that single cells actually are elongated on the groove. Let them increase in density, they lose the elongation. Then what we did an experiment here where we took these cells that had lost their elongation and that had secreted the basement membrane proteins. We removed the cells and we cultured new cells on the basement membrane protein that was secreted by these cells. And you see these cells are a lot less elongated than these cells, even though it's the same amount of time. This just shows us that proteins that were released by the cell here are basically what the cells are responding to. And then this is a genetic population of the cells where you can block the secretion of extracellular matrix protein or membrane proteins, and you see these cells are a lot less elongated than these cells. Here is the secretion of extracellular matrix proteins, cell density, masks the grooves, cell contact guidance. So the story is at low density, I've got the mechanism of contact guidance in these cells is the physical constraints on cell protrusions, and then at higher density I have a loss of the contact guidance response due to this released or secreted extracellular or basement membrane, the effect of the grooves. Okay. The other thing we've been doing with these, so at one point we started looking at cells live, right? So everything I showed you so far was on fixed cells, you know. So then we wanted to actually take some look at what happens live. And so we cultured cells on grooves, and we started, we labeled the nuclei of these cells, and we just imaged the nuclei. And this is on the left here, the nuclei moving around on a flat surface. And then this is what the movement looks like on these grooves, right? And actually, what we immediately were able to find, or to see, is that on these grooves, the movement of the cells is a lot more directional, right? They're primarily moving in the direction of the grooves. Whereas on a flat surface, they move in all sorts of directions. We started looking at this a little more carefully. So these are just another 2 movies. And if you look carefully at these movies, you'll see on the flat surface, they're moving kind of in all directions. Here, they're moving primarily in the X direction. But more than that, they're actually, you see kind of corridors of cells moving to the right and others moving to the left, right? You see this anti-parallel streams of you know, cells moving to the right, and then so we got intrigued by this and wanted to try and understand it.
Excuse me, why do they move?
That's a very good question. I mean, cells are actually never stationary. Cells are always moving. They're always probing their environment. They're always probing their environment. But they move differently if they're single cells versus collective. And so this is a form of collective migration. And the key thing here is these grooves are causing a type of collective migration that had never been seen before, these anti-parallel streams. Yeah.
No additional flow.
No flow. No flow. This is spontaneous that takes the form of these parallel streams. Very good question. Yeah, so I mean, what is right? So these streams don't end up extending the whole length of the thing, right? You typically have a stream that goes for, and you know, breaks down, and then something else happens, and then the cells move. But these organizations, collective movement, they always get these streams, but the stream might be going this way here, and then another one might be going here, and then there's a little bit of mixing, and yeah.
The mean flow is nil.
There's zero. Oh yeah. I have to conserve mass. So that's right. So this some trajectories that you accumulate over time on a flat surface. You see they're going everywhere. On the grooves, it's clearly these anti-parallel streams, right? And you can see kind of the emergence. This is the X component of the speed of these cells on the flat surface and on the streams. You can clearly see it. It turns out that the sizes of these streams, both their width and their length, are primarily determined by the depth of the grooves. If you go deeper, you get narrower streams and longer streams. And these are basically the velocity profiles. In here, you can see how you get, as you might expect, this periodic velocity profile because of these anti-parallel streams. So the cell streams are longer but narrower on deeper grooves. This is a form of collective migration because you don't see it in single cells. So if you start with single cells, they can go in any direction. It's only the streams only develop when the cell density becomes sufficiently high and the cells behave collectively. Okay, I'll just skip that. The other thing that we found is that these streams require junctions between cells. So if we disrupt the junctions, you can eliminate the streams. And again, further demonstrate the collective migration. So disruption of cell junctions induces loss of cell streams. Then,
can you tell me what's a cell junction?
Oh yeah, so junctions are basically, these cells, these endothelial cells, let's say you plate them as individual cells. They start proliferating, start dividing, and eventually they start making contact. And when they make contact, they form junctions between individual cells. And those junctions actually have just a complex of proteins. There are things like there are different types of junctions. There are gap junctions and adherence junctions and tight junctions. And each junction has a various, family of proteins associated with it. But it's basically links between cells. And it's that allows the endothelium to regulate its permeability. Because these junctions can be very tight and they don't allow things to go across very easily.
When the flow is over,
yeah,
then the junction is obliged to be broken.
It isn't broken. What happens is I guess in a way it's broken. It's between two cells you can get relative motion, and so that junction, it's broken, but then you form another one with the next one, and then you form another one with the next one. And that process is not well understood, how those junctions actually remodel under flow, or even without flow. I mean, they're always moving around, these cells, and forming, they're very promiscuous, right? One neighbor, and then one partner, and then another partner, and then another partner.
Sorry, and how you are breaking junctions?
We add a chemical called EDTA that basically has the effect of and calcium is essential for the formation of junctions.
So these junctions are mineralized.
Mineralized meaning?
There is some calcium compound.
Yeah.
So this is the reason for which we talk about calcification of arteries.
Exactly. I mean, you have calcium becomes, and not all plaques are calcified. Some plaques are very soft, but and that goes back to this vulnerable plaque business that I was talking about. It turns out that the calcified plaques are actually not as bad as the soft plaques that can rupture. Okay, so the other thing we tried to do in this study is we wanted to see if we can understand the physics behind this anti-parallel streams. So we put together a model that basically treats the cell monolayer as an active fluid. So an active fluid where the cells are treated as pneumatic active particles. Okay, and then the model says the way I'm going to include the grooves, I'm not going to include the grooves explicitly. The way I'm going to include the grooves, I'm going to say the grooves themselves play the role of orienting the cells. So it's just a constraint, an energetic constraint on cell orientation. And if I do, and this paper has all the detailed degree math that goes with it, I end up predicting basically this periodic movement of the cells, so the anti-parallel streams. And I end up basically, the model is shown in dashes here and the experiments in dots. And so the model ends up predicting the sizes of the streams, also the width and the length of the streams. Now, interesting thing is, if you think of this model basically says the effect of the grooves is simply a constraint on orientation. So if you follow that logic, that means any other stimulus that gives you a constraint on orientation should also give you streams, right? Well, one factor that can orient cells is flow. So we thought, okay, what about flow? Does flow? So we went back to the flow studies, and this is a movie now of endothelial cells under flow, and if you look carefully, you see, in fact, these anti-parallel streams. I mean, we've quantified this. I'll just play it again. And this is the quantification for the x velocity, and you can see you get these anti-parallel, just like you do with, this is not on grooves, right? This is on flat surfaces, but under flow. So just kind of, it supports the idea that an orienting stimulus is enough to give you these anti-parallel streams.
What is the order of magnitude of this velocity? 10 to the 6 × 10⁻⁴
per second. We have it in the paper, but it's 10 to the minus 2 microns per second.
And the images that you're showing, so the white part is the cell
border,
and then the dark part
is the cytoplasm, the inside of the cell.
Here, of course, you're not seeing the nucleus.
That's right. So we're going to talk about the nucleus in a second.
The length of these
streams
is related with the
depth of the groove.
The depth at the depth,
the depth, not the width so much.
Between.
Not so much the width. The depth of the groove is the primary driver.
So it's not a characteristic length of the cell. It is something related to the surface. Okay.
And shear stress that you say is there,
2 pascals.
What is that?
That means this is Poiseuille-flow. And so if I take, the Poiseuille-flow wall shear stress, which then relates basically, this is in a flow chamber, right? In a parallel plate flow chamber. So I've got Poiseuille-flow. So, if I know the flow rate, I know the viscosity of my fluid, I know the spacing, I know the wall shear stress. So that's the shear stress that the cells see.
And for the blood, what is the velocity of the blood?
So velocity is, of course, is one thing, but it's probably the shear stress that's most important, right? So the peak velocity in large arteries is 1 meter per second in the large arteries. That's the peak velocity because cycle, right? Meters per second. Yeah. And the shear stress is something like 1 to 2 pascals.
So the velocity of the cell is much slower than the...
Much slower, exactly. This thing is crawling on the surface, right? And the bloods go...
Okay.
Yeah. Yes?
Just very quick. Since here there are no points, what is determining the size of the streams?
So that's actually a very good point. I mean, and that's the model, basically. What it says is I have friction with the substrate. I have contractility by the cells that are pulling, right? And there is a balance that actually occurs between friction with the substrate and the pulling, the active pulling of the cells, and what end up emerging as the characteristic length is that. So it turns out after we did this study, we went back to some literature, and we found that these anti-parallel streams can be seen in other systems that orient things. So, for example, these are epithelial cells cultured on collagen fibers. There's this work here that had reported streams. There's, at the scale of inside cells, these are microtubules that are aligned by external magnetic fields. You kind of get also microtubules moving in anti-parallel directions. You also get what people have called laning pattern with high anisotropic friction. So we think it's kind of a more universal phenomenon where if you have cue or stimulus, you can start getting these anti-parallel movements. Okay, I'm just going to wrap up fairly quickly here with the last thing we've done on these grooves is when we started looking at the cells more carefully, we found that the cells deform a lot on these grooves. And this is kind of illustrated here. So each blue thing here is the nucleus of a cell. And then in red is the cell membrane. And you see, I mean, these cells are almost fluid-like, right? So they're kind of coating the surface. And if you then isolate the nuclei alone, you see how much deformation the nuclei are actually undergoing on these surfaces. You can get to a point where a nucleus can go completely become what we're calling caged inside these grooves. We've found different configurations of nuclei. This is a caged nucleus. This is a partly caged nucleus. This is a half-caged nucleus. These are confocal microscopy images. This is what we're calling a double-caged nucleus, and this is a suspended or free nucleus. So you get all these configurations. And so we've developed some algorithms that do the automatic segmentation of the nuclei and their quantification shape as well as changes. See, if you look at fluorescence intensity, also changes. So you can use information on shape and fluorescence intensity to basically categorize or classify the different types of nuclei. And we're feeding this into AI algorithms, right, or machine learning algorithms to kind of classify, do very rapidly, do from images, do classification of the types of nuclear deformations that we have. And so you can see, I'll skip this. The other thing about these nuclear deformations is if you look at things dynamically, you find that the cells don't, if they're caged, they're not constantly caged. They can actually be in the groove for a while and then get out of the groove. And this, for example, you see these two nuclei that are labeled with the arrowheads, like this nucleus. Here it's caged, it's elongated, and then towards the end of the movie, it gets out. Same thing with the yellow one. See, here it's caged, it's highly elongated, and then it gets out, right?
So the nuclei, with the size of the nuclei compared to the size of the cell is maybe about 10?
A nucleus is about 10 microns. A cell is 30 in diameter. But, exactly circular, but roughly.
Excuse me.
Yeah.
This is the effect of gravity. I mean, if you put the sample
No.
vertically, it will be the same.
Yeah. Gravity plays no role here, compared to the forces that are being generated by the cell, the active forces. The other thing that's really interesting is you can do AFM, atomic force microscopy recordings, and what you find on these nuclei, and what you find is the cells generally, the Young's modulus, the cells on the grooves are softer surfaces. You don't understand why, but they are. But the other more interesting thing is as the nuclei go into the grooves and get out, you see a sudden change in cell stiffness, right? So this is, and typically it's softening. So as you go right before caging to a caged nucleus, you get softening. Some cells do the opposite, but the vast majority that we've measured so far, as they go from caging to no caging or from no caging to caging, they soften. So the other thing we've gotten really interested in, that we found this really interesting in nuclear deformations, because there are some diseases that are characterized by abnormalities in nuclear mechanics. There's a class of rare genetic diseases called laminopathies that have actually mutations in a gene called the lamin, which is a gene of the nuclear envelope of cells, that the hallmark of laminopathies is abnormal mechanical properties of the nuclei. So these laminopathies involve mutations of the lamin gene. They involve abnormalities in nuclear structure and mechanics. There are 5 groups: muscular dystrophy, neuropathy, cardiomyopathy, lipodystrophy, and progeria, or premature aging. Mortality rate, they're rare diseases, but they, and there's no cure, right? So we thought it would be interesting to see if nuclei from healthy cells versus diseased cells would deform differently on our surfaces, on our grooves. And sure enough, they do. So we struck a collaboration with Catherine Coirot, medical biology. So this is on flat surfaces. These are what the healthy cells look like. And so Catherine Coirot gives us cells from patients, from laminopathy patients. So these are healthy cells and these are diseased cells. And then, so you can characterize cell shape, for example, the roundness or the circularity. And you see on the flat surfaces, the healthy cells, this is distributions, right? The healthy cells and the diseased cells showed similar distributions. If you put them on the grooves, then that amplifies the differences, right? So you see these cells now, the disease cells, deform a lot more on the grooves than the healthy cells. And so we think this could be, we think this could be a tool to basically diagnose these, very easy and simple and cheap and high throughput tool to diagnose laminopathies. And that's, we're going in that direction now. We just a patent on this, and one of the postdocs is interested in pursuing this. So anyway, so I think that's all I wanted to mention. But before I wrap up, Yves mentioned the Beltrami Prize, and I just wanted to say a few words about how deeply honored I am to receive this prize. Thank you very much. And I I wanted to say maybe a few words about Eugenio Beltrami, who was born in 1835 on November 16 in Cremona, in Lombardy, and died on the 18th of February 1900 in Rome. And this was kind of his professional CV here. He studied applied mathematics at the University of Pavia from 1853 to 1856. And then he ran out of money and worked for a while. So he worked for 5 years as a secretary to a railway engineer in Verona, sorry, missing an E, and then in Milan. In 1862, he published his first paper and was appointed as a visiting professor of algebra and analytic geometry at the University of Bologna. Stayed there for 2 years and then became the chair of geodesy at the University of Pisa. From there, moved to the University of Bologna, stayed there for a few years, then moved as chair of rational mechanics at the University of Rome, stayed there for a few years. So he moved quite a bit, actually, and then went back to Pavia and was chair of mathematical physics for 15 years, and then went back to Rome, and where he ended his career. But so most of us probably know about his work on curves, on surfaces, and on non-Euclidean geometry, but he actually did a lot more than that, and this is something I did not know about and learned about. He worked on optics, on thermodynamics, on elasticity, and magnetism, and he really published seminal work in these various areas. So I think Italians are very good at producing these amazing scientists who are just true Renaissance men who happen to work on everything and are able to do. And he worked on Maxwell's equations. He even worked a little bit on the history of mathematics. So really an amazing contributions. But this is a quote from Beltrami that I found really interesting. I'm just going to read it here. He said, "Students should learn to study at an early stage the great works of the great masters instead of making their minds sterile through the everlasting exercises of college, which are of no use whatever, except to produce a new Arcadia where indolence is veiled under the form of useless activity." And then he said, "Hard study on the great models has ever brought out the strong, and of such must be our new scientific generation if it is to be worthy of the era to which it is born and of the struggles to which it is destined." So with that, I think this says it all. Thank you so much for taking the time to listen to this, and thank you so much for the honor of the Beltrami Prize, and thank you for your attention.
I would like now thank you for your huge presentation, of course. It's my honor and my pleasure to give you these medals of the Beltrami Prize. We understand now what is vascular fluid mechanics and very extremely nice results, experimental results, which are the basics of the science, of course. And I have also the pleasure to give you the diploma of this prize on behalf of the International Center of Mathematics and Mechanics of Complex Systems. Yes.
Thank you very much. Thank you very much.
Less serious comment. Do you have remarked that only old people were very interested in this presentation?
Atherosclerosis, I mean.
But anyway, thanks for doing this job.
Thank you.
Work harder, please.
I'll try.
You are lazy in the last speech.
I know. I'll try.
Congratulations.
Thank you.
And,
Thank you.
Of course, we can have maybe a discussion with you. I have a first question.
Sure.
Please don't take my question as a complete personal.
But it is.
What is the effect of alcohol?
So actually, it's interesting. So the risk for atherosclerosis, actually, I talked about cholesterol, right? Cholesterol is carried by a molecule called low density in our body. So that's what we call the bad cholesterol, right? But there's also another molecule called HDL, high density lipoprotein, which is what we call the good cholesterol, and actually removes the bad cholesterol from your body. So your risk for atherosclerosis is primarily driven by the ratio of HDL to LDL. So not so much by LDL levels alone or HDL. Now going back to your question about alcohol, it turns out that LDL you can control somewhat with diet, but it has a huge genetic component. HDL, one thing that does improve it a bit is a little bit of alcohol. Okay. So there is some data that suggests that a bit of alcohol can raise HDL levels, and that's a good thing. But then there's more recent data that suggests alcohol is bad from perspective of cancer. Yeah, okay. So, you know, I'm not suggesting go out and drink, right? But,
but wine is not alcohol. just one more, please, and more serious, really. We know that for instance, for MSc, the development of cells depends not only on the geometry of the substrate, yeah, but also on the Young's modulus, yeah, of the substrate. Yeah. You never, yeah, talk about that.
That is true.
What your comment was?
Yeah, that's true. The rigidity of the substrate is also a very important determinant of cell shape. And ultimately, in real life, all of these together, the flow, you've got the topography of the surface, and you've got the rigidity of the surface, right? We haven't looked at the rigidity aspect, but it would be an important thing to also incorporate.
Okay, I cannot... okay, sorry.
You can ask me your question.
Go ahead.
One curiosity, when you show the microgroups,
yeah,
yeah, in my mind, yeah, there was a concept similar I found in the literature concerning wettability and adhesion.
Yeah.
So, can you comment, yeah, if you found some relationship?
Yeah.
Because I found some similarities with the wettability of the fluid.
You're absolutely right. Actually, this is something that we've gotten very interested in. You know, especially there are people who look at droplets and how they interact with grooves, right? And there are different regimes. You know, in some cases the droplet remains completely suspended. Then there's the Cassie-Baxter regime where you get full wetting, you know, which looks like our caged nuclei, right, where the cell surface. So we're lucky to have one of the world experts in these wetting phenomena at École Polytechnique and in Paris, David Carey, yeah, and he and I are actually beginning to collaborate on this notion of the analogy between droplets and cells, and to what extent I mean, the analogy is valid to some level, but not completely, because these cells are active, right? So they're producing their own, generating their own forces. But it's really interesting to try and separate out what might be a passive wetting phenomenon and what might be, you know, the contribution of the active component.
I would simply wanted to comment, especially for young people, that in the present extreme specialization of science, now our prize winner is neither a doctor nor a fluid mechanician, so he could have serious troubles to get a position in Italy. And what is astonishing is that French system did manage to appreciate and give value to this kind of profile, which is the Beltrami profile, right?
Yeah.
We invented this interdisciplinary criterion, but now we are in the cage of disciplinary groups.
That's a very good point. In fact, you know, but also in France, I think we talk about wanting interdisciplinary science, but ultimately, when it comes to awarding positions, very often people fall on the side of disciplinary stuff. And Yves done an amazing job trying to educate CNRS as to the importance of biomechanics, for example, as opposed to just, so, you know, only fluid mechanics, only solid mechanics, as, you know, let's, so, and I think he's continuing to do that.
Umberto Eco said sometimes that God, if he applies for a position in Italy, will not get it, because at the end, he wrote only one paper and he was talking about everything.
That's a good point.
Anyway.
Yep.
So I have a couple of questions, maybe one comment about what Francesco said. These cells are always mobile, so how do you catch them to do the AFM on the cell? So that's one question. The other question was, what kind of substrates are you using at the moment?
We're using PDMS substrates at the moment.
Coat them, or these are just PDMS?
PDMS grooves coated with fibronectin. And then in terms of moving, I mean, they don't move at the rate, you know, it takes us a few minutes to make an AFM scan, and during that time they're still there. I mean, you know, they move at 10 microns an hour, and a cell is 30 microns, so they're moving a third of their body length an hour.
Okay.
So
So the comment about the previous comment. So without discipline, we would not have interdisciplinary, right? But these days in the US, I'm sure you are aware, there is the push on convergence research. But at the end of convergence research, they want new disciplines to emerge. So,
no, but it's, I mean, you know, it's a challenge, especially for young people today, who are interested in working on these areas, to, you know, what is the best way to sell yourself? But, you know, one thing I think Beltrami said here is hard study of great models that's one thing that's very important. The other thing he said that I find amazing is he says through the everlasting exercise of college, which are of no use whatever, right? So, you know, it makes us really think, and this was, you know, back in the late 1800s that he said this. So are we preparing our children and our kids today for, you know, the best, for the future careers that they're actually going to be seeing, you know? Are we teaching them the right way? That's, I think, one thing we need to be thinking about.
Okay, thank you so much, Professor.
Thanks.